Hello everyone, I am Dr. Dheeraj. Today I am going to talk about chromosome. So this chroma means colored and soma means body. So this is colored body uh, present inside the cell. So this chromosome is very interesting and important component of cell. And we all know that these chromosomes basically carry the genetic information from one generation to another generation. So genetic material basically is chromosome. So we will see how these chromosomes are formed and what is the real meaning and significance of this chromosome. So in this particular video, I'm going to talk about the detailed structure of chromosome and the chromatin. So this is the structure of interface cell where one can observe a thread-like structure inside the nucleus, and these thread-like structures are known as chromatin reticulum. But these thread-like structure forms chromosome during cell division. So this is two important phase that if the cell is in interphase, one can observe chromatin fibers, and in M phase, one can observe is condensed structure that is chromosome. So somehow there is a relationship between these chromatin and chromosomes. So basically, chemically, these chromosome and chromatins are almost same, and this is the condensation which is responsible for the conversion of this chromatin into chromosomes. So if we talk about human cell, like human cell contains 46 chromatins fiber, and each chromatin fibers contain single DNA. So if we observe the chromatin fibers in interface, one can observe 46 chromatin fibers, but in cell division, one can observe chromosome not chromatin fibers. So again, 46 chromosome can be observed, but they are highly condensed, they are thicker, and they are smaller in size. But the chromatin fibers are longer, they are thread-like, and they are less condensed. So this is the condensation which differentiate between chromatin and chromosomes. So this chromatin interface, they exist in the form of chromatin, and this space is very important where DNA replication takes place. So if DNA replication has completed, each chromatin fiber will contain two DNA molecules, and if DNA molecules are double, so in this M phase, double stranded chromosomes can be observed. So now from this discussion, it is clear that in interphase, the term chromatin fiber will be used, and in M phase, when the condensation is very high, chromosome term is used. So chemically, they are same like chromosome and chromatin. They are made up of DNA, histone protein, non-histone protein plus RNA. So now we will see the detailed difference between chromatin and chromosome. So if we see the chemical nature of chromatin fiber, these fibers are made up of DNA, histone protein plus RNA, and non-histone protein. And we all know that these chromatin fibers are present during interphase, and interphase is transcriptionally very active phase. So the RNA, whatever RNA will be synthesized during interphase, they sometimes they remain associated uh, with the chromatin fiber, and therefore this RNA is also included with the structure of chromatin fiber. But we all know that after condensation, transcription is inhibited, and RNA will not be produced. So in case of chromosome, RNA cannot be observed along with the structure of chromosome. So chemically, the chromosomes are made up of DNA, histone protein plus non-histone proteins, but one cannot observe RNA. So this chromatin fiber is thread-like, less condensed structure, and when the condensation increases, they form chromosomes. So as we have discussed, if this chromatin is before S phase, it has only one DNA, but after S phase, each chromatin will include two DNA molecules. So the in double-stranded chromosome, like two DNA molecule can be observed, or it includes two DNA molecules. And this is double-stranded chromosome, and the arms of chromosomes are known as chromatids, and on the surface of chromosome there is depression known as centromere. This centromere also include other protein like kinetochore protein, which is important for cells division. So these chromosomes, when formed, they are highly condensed, and maximum condensed chromosome can be observed during M phase of cell cycle. And again, if decondensation takes place, these chromosomes will be converted into chromatin fiber, and again after condensation, the chromatin fiber will be converted into chromosomes. So this cycle of condensation and decondensation occur during cell cycle and cell division. So at the time of interphase, DNA replication takes place, and when M phase starts, condensation of chromatin fiber also starts, and after completion of M phase, decondensation of chromosome takes place, and cell is converted into, again cell is enters into interphase. So at molecular level, these chromatin fibers are made up of DNA, histone proteins. They are the major component. And first step, they form a spool or core particle where four molecules of different histone proteins combine and form the tetramer of histone. And when the dimerization of these histone protein takes place, they form octamer of histone, and this octamer of histone is known as a spool or core particle. And when the DNA is wrapped around this core particle, the structure is known as nucleosome, and this is a structural unit of chromatin fiber. So when different chromatin fibers, nucleosomes are associated, it forms a chromatin thread, and this is a 10 nanometer fiber, and these nucleosomes are associated with each other with the help of H1 histone, which is also known as linker histone. So this histone fiber or histone thread, when it is condensed, it form chromosomes, and when chromosome is decondensed, the chromatin fiber structure is restored. Thank you.